Hello, Wataru.
Hello,
How are you?
I'm fine. And
Good.
you?
Yeah, pretty good. Pretty good. All right. So that's great. How are things going?
Yeah, um, I'm actually a little bit busy by the medical training.
Okay.
Actually,
Yeah.
I'm a medical student, so yeah, the medical training is very hard now.
Yeah. Yeah. So yeah, we met on Thursday about the GSoC project. How are you finding it?
Um, I was asked by Jiahang to check the data set, check the contents of the data sets,
Okay.
such as epic.
Okay.
Let me share.
Okay.
Here it is.
Oh, okay.
This Notion page.
Yeah.
The epic data sets, and this is the cell tracking challenge datasets, and C2, C12 datasets, and worm image datasets. This is provided by you.
Yeah. Yeah.
Yeah. And the result was epic dataset only contains fluorescent movies.
Yes.
So no annotations.
No.
And cell tracking challenge. Many of cell tracking challenge datasets have manually generated segmentations.
Yeah.
Also, yeah, the datasets of C. elegans in cell tracking challenge didn't contain the tracking tracking labels.
Yeah.
I mean, which are the same as the ones in the next image. Yeah.
Okay. Yeah.
And C2, C12 dataset had the true segmentations and true cell tracking annotations. So this can be this contains the lineage tree.
Right.
Yeah. Yeah, that's all.
All right, that's great. And then, yeah, worm image dataset with segmentation. So, what are you planning on doing with the? Like, I know that the labels are important. You have the cell. I guess you have some data in different data sets. You have different types of data, and you're going to put them together, or are you going to use all of the data sets in different ways, or what are your plans for that?
Actually, my plan is to search for interesting GNNs.
All right.
And I will then decide which dataset to use. Actually, I'm thinking like that.
Okay. Yeah. Yeah.
What do you think?
I think so. Yeah. I think you should treat every data set as a potential source of information. So some of the data sets are going to, when you start to go and try to segment them, some are going to have certain types of information highlighted. Others are going to have other types of information highlighted. So like when we did our meeting on Thursday, we talked about, thinking about the source data and how it can affect like what you get at the end. So you're processing the data, you segment it, and then you get this network, but it's contingent upon what you're able to extract out of the data sets. And so like something like a fluorescent data set is going to allow you to extract certain features more easily than other data sets. Now, in the past, we've had this same issue where we've used different algorithms to segment different types of data. So, if you have a really nice high resolution image, you can use an algorithm, and it can pretty easily pick out a lot of the features that you want to use to segment individual cells. So you can segment the nucleus, you can segment the membrane, and you can segment some other things. In fluorescent images, you have just kind of like the center of the cell. You have basically the nucleus where the fluorescent protein gets expressed, and then you may have some area around it. And there are also things like autofluorescence, which is where you have this noise. The fluorescence is expressed where it shines in that signal range due to noise. And so that's something that usually they clean up I don't know how these data sets are released. Usually they're cleaned up in the images, but not always. And so these are all things you have to keep in mind. But I think like the lesson here, you have a nice selection of data sets to go through. I would try to like just work with some of them, maybe extract some networks, and then look and see if they're comparable. Do you have networks that are sort of the same, or network embeddings that are the same or similar across data sets? If they are, then it's likely that that's a real signal. If you get a lot of embeddings that are incomplete, or they're they're all over the place in terms of what they look like they don't look like each other at all, then that might be a problem. But I don't think that'll be the case, because the data, the reason these data sets are released, they have some quality control. Not really sure what the quality control is on each data set, but I think you can safely assume that they're workable. You're able to work with them, and then you're able to extract these. You'll have to find. Usually we use the nucleus as a marker. We use some centroid. We call it a centroid because we don't want to call it the nucleus, because it's not really the nucleus per se. But you would use something like that fluorescent signal. That would be the centroid of a cell, and then your embeddings would be some criterion on those dots, which would be the nodes. So it could be distance, like we had mentioned in the meeting. It could be some other criterion. But I think the important thing here is to extract the data, to segment the images. You may have problems with error, like false positives or things like that, but that's something that you can control. It's something that we can control. We know how to do that. So just to keep those things in mind, that this step is actually pretty key to like getting these correct network embeddings. So yeah, this is nice. Okay.
Thank you for the advice.
Yeah, thank you. Thank you for showing that. That's nice. And I like the fact that you're using Notion. I know that Jesse Parent is a big fan of Notion, so it's nice to see it being used like this. All right. Thank you, Wataru, for the update.
Thank you.
So we have a new person joining us, Longhui Jiang. Do you want to introduce yourself? And you can type in the chat if you want.
Oh, hi, Bradly.
Oh, hi, how are you?
Hi, just like what I said, we have a new guy join us.
Okay.
He called Longhui, and he's my university classmate, and I think he is proficient in the PyTorch, Python, and some other deep learning tools. And I've described some details of our research team, and he has some experience on it. And I think he is very interested in joining such a project and our research idea. And so I think because his experience in such as computer vision and such as some other deep learning tasks, so I think probably he's very helpful for us, and he could help us to build a better GSoC project. Yes.
Okay.
And Longhui, could you please briefly introduce yourself?
Hello. Can you hear me?
Yes.
Yes. Maybe this microphone is noise. I hope my voice is clear.
Oh, yeah, it's clear. Yeah.
Hello, I'm Jiahang Li's friend, and I'm very interested in this project. It's about GNN. Yeah. And I think I can do something to make progress. Yeah.
Yeah. Well, that's good. Yeah, I know Jiahang told me that you were going to join us. He's going to join in with him and Wataru and join the meetings and that sort of thing. So welcome to the meeting.
Yeah. Thank you.
Yeah. And we're giving updates on different GSoC projects. So Wataru started with some of his work on, extracting data from data sets, going through the data sets, understanding what they are, and then, there's some quality control, and then, there's some extraction of data, some segmentation. And so all of that should be, the sort of the key to this project, because you have to have good starting data to get to the embeddings part and to some of the other things you want to do. So, I was really impressed with the meeting on Thursday. I think that there's a lot of good vision for this and a lot of, potential. And so we'll see how we can progress through the summer here. Jiahang, did you want to give an update on your work or?
Probably as for this week, my update is about the last small meeting between me, you, and Wataru. Remember about that GNN idea and cell tracking? Yes, and I'm still doing some research on some downstream task of this cell tracking idea. Like what I said before, we need to design an appropriate task for our research idea, because by now we think probably the link prediction of graph neural network is a good choice, but we have multiple options, such as I think the multi-object tracking or some other ideas related to us can give us some inspirations. So I'm still reading some related papers, yes, and at the same time I'm still implementing some structure, some graph neural network structure, which is actually the stage 3 of our GSoC projects. Yes, it should be the update of this week. Yeah.
Okay. Yeah, that's good. And so Wataru mentioned about the cell tracking or the cell tracking data, and I don't know what this. I mean, one of the data sets has it, but it isn't like directly relevant. I mean, some of this too. When I say quality control, what I mean is like matching up some of the different data sets. So like if there's cell tracking data that you have, making sure that it makes sense with respect to the sort of embeddings that you get or the segmentations that you get. So that's some of that is just kind of like you're just going to have to check it, like, plot it out or see what it looks like. But, that's something, and I don't know, every time you take data from a different source, there's always a potential for like error or like, a mismatch of things, because not every group uses the same methodology to get to the output data. So sometimes cell tracking in C. elegans, it's rather easy because it's relatively easy because there isn't a lot of variation. We know what the cells are and what their tracks, kind of what their final positions probably are. So the tracks are usually correct if you go from one data set to another. But I'd still check everything to make sure it works and that it makes sense.
Yes. Yeah, I understand that. Yes, I think maybe we will start with trying C. elegans and just make a demo so that we will make sure that all things just work like what we imagine, and then we will move to other large data sets such as cell tracking challenge or such as some other data set that we use in the DevoWorm. Yes, but we will start with the C. elegans because I think it is small and simple. Yes.
Yeah, that should work. And then you can Okay.
Yes, and just like what I said, I've mentioned the paper last week, and I think they have some source codes which will be helpful for us, and I will try those source codes because they also deal with those large data sets, such as those cell tracking challenge data sets, and I will check how they implement their codes to deal with those large data sets to segment them or to extract positions of these cells. Yes, I will check their source codes.
Yeah, that's good. Yeah. Okay, well, thank you, Jiahang. It was a nice update. Okay. So next up, Karan or Harikrishna. I'm going to switch gears to a different project here. Are you there, Karan? Looks like you're unmuted.
Okay. Now I'm audible.
All right. Yeah, you're audible.
I think, I'll start sharing my screen.
Okay.
Yeah. So my week, like I outlined, I think in the previous update, that I was improving the contour extraction for the initial stage of building the 3D model from the 2D image. So for this particular one, this is like so far how we're going about, if you can see, these outlines, how they're mapping about this axis of rotation. So this is again, just a linear programming algorithm that I designed, based on this. So what it's doing is it's taking 2D images, taking their outlines and their centroids as well. And based on the centroids of all these stacks of 2D images, it's trying to build the frame. So this was the, linear programming way of getting to the answer. The things that, Harikrishna also told you about, that we were trying CoalMap and, neural radiance fields for improving this particular step. Mm-hmm. So the thing is, there's still, a bit of a learning curve to get to do that. So that's why even Harikrishna was having, issues in rendering a model using, like improving the current techniques that we are using, just, going a step further than the linear programming way. Otherwise, for the next week, I'll be, improving this further by extrapolating more outlines based on the existing ones that we have there. So based on this, for this week, I had to calculate the axis of rotation and the angle as well. So far it looks okayish, but, I'll have to tweak, come back to the step again after I, get the final model out again.
Right. So what is this what we're looking at? Like, is this like you're extrapolating different?
Yeah, I'll go through it. I'll go through the previous steps first.
Okay.
Yeah. So these are the outlines, that we had gotten from each of the 2D images that we had.
Yeah.
These are not all of them, I think. Yeah. So from the data set that I was doing, like for that particular image set, the angles were like 20 degree, 35 degree, 50 degree, like how much they're rotating by each image set. Like the first image and the second image, the angle of rotation between those two images is what we were approximating to, transpose these frames here.
Yeah.
So this is like the early rendition way of, of getting better at approximating the final frame of the 3D model. So this was slightly improved because earlier, I had to resize these outlines along the common axis because some of the images, they have the axolotl subject, slightly far away or slightly close to it. Like you can see, still there are these two outlines, that are kind of still not resized properly. I think one of them is not resized properly. So the thing is to, I'll be increasing the number of outlines so that I can create a mesh from this using the existing outlines that are there. Otherwise, yeah, images, no, these are the other images.
Okay, so those are the source images. And so those spiral, it almost looks like a spiral, kind of like that. Those are just these.
No. Yeah, this transformation technique,
Oh, okay, yeah.
It's mostly this so far, yeah.
All right. So these circles, kind of in a spiral or whatever, they're just these 2D slices that are aligned in different angles.
For the frame, yeah.
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 And so you hope to build like a 3-dimensional model based on those planes, or?
Yeah. So I'll be improving I'll have to come back to this step unless I can see how the final mesh is forming from the base that is this. So I'll be, like I told you about the function, of getting more lines based on the existing one. So I'll see how that works out. Otherwise, we'll keep on, trying to try out new things like whole map anemia. But even this will, give a very close approximation. So I'll keep on doing this by the side and think of ways to improve it.
Yeah.
It can be done. Yeah.
Yeah.
Do you need any more images to work from?
Yeah. The recent ones that were shared on Sync, I think we have those. But yeah, any more images per the sample, like if you're clicking around 20, 30, I think these are 8 or 9 images for this particular axolotl and there. So
okay, I put some up on Sync.
Yeah, I know. Harikrishna.
You know about that? Were you able to access them at all?
Yeah, I access them, but I've not tried using them with the.
Oh, okay. Just so long as you do have them.
Yeah. Okay. I'll try those as well. Yeah.
Yeah.
So this is just one axolotl sample data set. This is I think 8-9 images for that. So this will have to be replicated for that one. This is scaling the algorithm that we currently use. That's the thing. Yeah.
Well, that looks really good. Thank you.
Okay. Thanks, Bradly.
Harikrishna, you had an update? Hello?
Yeah, hello.
Hi. All right.
Yeah. So right now, till now it has failed. So last week what I did was I was reading some other methods, and parallelly I started building the UI of the application, of the final application. So yeah, I'll try finding new ways, or else I'll stick back to old methods.
Yeah. So you said that you had some problems.
There's much improvement.
Okay. So you said you had some problems with some of the other stuff that you were doing, some of the techniques. Yes. So what was the problem exactly?
Yeah, so the problem was the algorithm takes certain images, many images as an input, and it wasn't able to give the result because it identifies the images as different objects. So that's because it doesn't understand that the images are continuous. It feels like the images are not continuous. So I tried some image augmentation with the data set, like rotating the images or translating the images in different directions so that I could increase the numbers, but still the result was the same. So either I will try doing something with the data sets again, or I will move on from this algorithm and try something else.
Okay.
Also I parallelly started building the UI of the final application.
Oh, that's good. Yeah. Now I like that you're moving on to something else when you're having trouble with it.
Yes.
Yeah. So what are the algorithms you're using?
The one which failed was called ColeMap. It was a neural network algorithm. But I'm not sure what I'll be moving on to. I'm just reading papers now. I'll figure it out within 1 or 2 days and continue with it.
Okay, yeah, that's good. I think trying to find out what the assumptions are of some of the algorithms is good. So if you can figure out what if you want to have a candidate algorithm and you want to see what it's best, what it does best, that's usually a key to where you can focus on actually implementing it.
Yeah.
So I mean go ahead, Susan.
I'm still working on putting some stripes on a small ball bearing in hopes that might help with some of this. Are we still interested in stripes on a small ball bearing?
Is this something that would augment the image, kind of like the boundaries of the image, or calibrating the algorithm?
Kind of calibrating it. I could take say an 8mm paintball ball, and it's slightly bigger, so it's easier to manipulate, and put some stripes on it. There's a couple of ways I was going to do that. So one was to get a mesh that I have hot and kind of melt the paintball ball with some stripes. I'll see how it works. I've had to take a week off, but I'm okay now, so I can maybe see about that.
Yeah. Well, yeah, would that work, Harikrishna? Do you think that might be helpful, or?
Yes, I think those images can be helpful for trying out.
Yeah. Okay. All right. Well, I'll try to get you some striped tiny balls. I have my paintball balls are yellow in color. So I don't know if that's the best color or not. There are other ones available. But I guess I could try it with the little yellow ones.
Yeah.
Okay.
That would be good.
Yeah, I guess that will be helpful.
Yeah.
All right.
Well, thank you, Harikrishna, for the update. That was very good. And I'm glad to see, some of the troubleshooting going on and some of the experimentation that people are doing with projects. It's very important to go through the project and to kind of hit those marks, where if you have trouble, if things go well, that's great. But a lot of times people are going to have obstacles that they need to overcome. So this is great that we're thinking this way and that we're keeping on top of this. So I don't know if anyone had anything they wanted. Here's Morgan. Hello, Morgan. I don't know if there's anything anyone wanted to talk about or present today, aside from GSoC updates. Okay. I wanted to say also, I know he left, but Wataru made a pull request on DevoWorm, and he made a small improvement to it, so it was accepted. Thank you, Wataru, for that. That was a small fix to the DevoWorm platform, and Jiahang and Wataru were working with that, and they found some problems with it that needed to be addressed. And that's really the essence of open source. Is you build something and you make it, you make it go live with version, the first version, and then you update it according to maybe new features, or sometimes you have to correct bugs in the software, or you have to improve it in some way. And so that's where people using it comes in, because people using it are going to be able to identify those problems and then make corrections on their own. So that's a nice demonstration of open source making a correction there. And I don't know, we'll probably be releasing a new version of DevoWorm at the end of this summer, after we get some graph embeddings and we can incorporate them. So that'll be fun. So I'm going to share my screen now. I'm going to go on to some things that I wanted to talk about. So let's see. All right, you can see my screen. Good. So we do have this list of major tasks. I always bring this up every once in a while to talk about some of the projects that we have ongoing in the group. It's pretty diverse. As you can see, there are a lot of things here, a lot of issues that are sort of unaddressed. We have some things that are done, finished. We have some things that are in progress. We have some action items. We have holds and to-dos. The to-dos are mainly things that we've come up with as ideas, and we're not really there yet. So some of these things are maybe things that have fallen through the cracks in hold and to-do, but there are things we can get to with, if someone's interested in it, they can go ahead and do that. So this is in the DevoWorm repository under group meetings, and it's the project. There's only one project board here. So this is major tasks for 2022. So this has been something we've been working on since 2020, kind of going through some of these things. So we have things like papers, and we have lectures that people might want to do. We have these different projects that, we will make a presentation somewhere, and then, we'll have follow up on that. And this is the place we put this. So, we have a lot of outstanding issues like pattern resumption after self-repair seashells. We've been pursuing that work for quite a while, actually. But we haven't really been able to realize it. This is a project where you take pictures of seashells, you take like the three-dimensional images. It's kind of like what Harikrishna and Karan are trying to do, where you take those images, you stitch them together into a sphere, and then, or into in this case, some sort of torus, and then you can view the thing and analyze it in this 360 view. Actually, once we get this work that Karan and Harikrishna are doing in place, we might be able to just get those images and do the whole thing automatically. So that might be good. We've also have an interest in working with virtual worlds and simulations. If you've not seen the OpenWorm viewer, you're invited to go look at the browser. It's browser.openworm.org. And that's an example of one of these virtual organisms, or how you can build a virtual organism. Basically, you have the worm, you can zoom in on different cells. The cells are annotated, and the whole thing is built in Blender, so you can manipulate the different parts. You can see it through a viewer and those sorts of things. I've been thinking about building not only an embryo in a virtual environment, but also some of the parts of cells and developmental processes. So this is something that I've had as issue number 113. If you're interested, contact me on that. There are also things like the Mathematics of DevoWorm document. This is actually something that is now made into a poster, so I may move this over later to in progress or finished, because we've kind of moved in, this is kind of like a coherent poster now, so that's good. We have a video highlight of this new paper that isn't quite out yet, but when it comes out as the book chapter, this bacillary aneural cognition paper, that'll be out as a video highlight. And we have a YouTube channel where we put different things out, so that'll be one of the things that come out. Maybe at the end of the summer, we can make video highlights of the different GSoC projects as well. That's something we can put on the list. So we have all these ideas, and they kind of they're kind of loose ends that get thrown out at the meetings, and you can see there are a lot of them here. So if you're interested, go through this list. If you see something you're interested in, contact me. You can put in addition to the issue, so you have the issue and you just make a comment, and I should get an alert about that. So I know that Morgan's here, and I'm glad that he's here, because I wanted him maybe to talk a little bit about some of the stuff that he's interested in with respect to organoids. We haven't talked about organoids in the group in a while.
Hi there. Yeah. So it is school run time here in San Francisco.
Yeah.
So I'm actually just getting some things together to get my daughter to school, but
Okay.
Organoids, certainly some of the self-organization of just stem cell work is it's been what we've been looking at recently, but not much new, not many updates. Sorry.
Oh, it's okay. Well, I just wanted people to be aware that you were interested in that.
Oh, sure.
Yeah. So
Yeah. Please.
Yeah.
Please.
Yeah. So we've done some stuff. We've talked about organoids in this group. There are neural organoids, which are where you have neural cells that you can grow up into these little organs or organoids. So you have like a bunch of neural stem cells, and you differentiate them into different types of neural cells, and you get these little structures that look like, no, they may not look like brains. They could be brain structures. And then you can test hypotheses about function. You can look at like disease states. You can do that, other sorts of things. And of course, they've done organoids for every different organ in the body. So you have liver organoids and, other types of organoids that are somewhat informative in terms of like what, is going on. It's, we have cell culture, and we say, well, cell culture is a good analogy for what we're doing in the body in terms of medicine. So if we have a plate of liver cells, we can sample those and say what the liver, maybe like metabolism in the liver, or we can talk about like gene expression in the liver or something like that. In organoids, you have this added dimension of phenotype. So you have this, first of all, usually there's a third dimension to the organoid, but also you have this function of the organ itself. So you start to get things like, different types of associations between cells and things like that. So, this is kind of the bridge between cell culture, where they grow cells in a dish, and, they can control it very tightly, and like animal models, and looking at the development of organs and whole embryos, which is what we do with some of the microscopy work that we're analyzing. Really, our goal there is to look at some of the things that are going on in that process. And so organoids bridge that gap. And so Morgan's been interested in trying to get some things off the ground with that. And I think, longer term, I see this as being kind of a promising area. There's also the idea of computationally modeling organoids. So, this is something that we've done with computational modeling of embryos. We've talked about physics modeling. We've talked about some of these other things. And organoids, we can also model organoids in terms of sort of the real early origins of cells self-organizing into different types of associations. Susan?
I might be interested in measuring an organoid.
Yeah.
Or two. I'm at the University of Manitoba, so I don't know where to go for them. I'm trying to do optical coherence elastography, and I'm doing modeling in COMSOL of cells, currently just epithelial cells, and I was kind of intrigued by organoids, partly because I think they have a difference in pressure between the outside and the inside of the sphere of the organoid.
Okay.
So, yeah.
Yeah, I can also say, in addition to the organoid work, that I've done a lot of finite element modeling too, which is what COMSOL is for. So yeah, that's great.
Yeah.
That's interesting. Have you done any tensegrity modeling with COMSOL?
Not tensegrity itself, although we have built some of the kind of small robots or small structures. So very super interested, absolutely. And that would be one of the things that I would be interested in doing if you're going to do finite element modeling for robotics, for sure.
Oh well, this is finite elements for cell structures.
Sure. We're treating them kind of interchangeably.
Well, yeah, they can be interchanged. I know.
Yeah. But that's great. I just saw just recently a Phoenix model, which is the finite element software that we use. A Phoenix model for a 3-sphere swimmer. So there's some other projects that have been looking at like motility, which I think is interesting too.
Yeah.
Oh well, yeah, I'm stuck with doing tissue.
No, that's awesome.
This is by order of my supervisor, who doesn't like my tangents. Yeah. Anyway.
Yeah.
I laugh. Yeah, I have too many tangents. Anyway.
Me too.
Yeah. Anyway. Thanks. I'm struggling with my model right now.
Which model was that?
The 3D tissue model.
Oh, okay.
It keeps telling me its set is empty and that it's unsolvable, which is typical of a tensegrity object, but then I don't know how to find the data that it's throwing, so I don't know where it is in the set of data that there's an empty line or whatever that it's going on about. So I'm having some trouble.
Oh. Well, that's a shame.
I'd like to actually see the output. I don't want to see a picture saying, oh yeah, it doesn't work. Like, that's so useful.
Yeah.
I need to actually see the matrices that it's throwing.
Right.
But COMSOL is, they say, oh, you don't want to see that. I'm going, well, I have to see that. Anyway, yeah.
Well, yeah, it's always a struggle with modeling. It always seems so easy, and then it's not. Yeah, so Jiahang had to leave, so thank you for attending, Jiahang, and it looks like Wataru also left. So yeah, why don't we go back? I have some papers to present, and I have a couple of things actually to talk about. So let me share my screen again. Thank you, Morgan and Susan, for your updates, and Harikrishna, thank you for attending. So this is, I have a couple things here to talk about today. First of all, Dick Gordon sent me some figures from, I think this is from one of his books on differentiation waves and differentiation trees. And so we were talking, I don't have the paper right now, but it's up on ResearchGate. It's a paper I'm going to be presenting on later this month at the Network Science Conference on embryo networks. A little bit different. They're hypergraphs that represents multiple features of development in what I consider to be a C. elegans-like embryo. And so I kind of walk through that in the paper. So it's a little bit different than the GSoC project, but it's related. Now, Dick sent me these pictures, and I think they're from his first book, the 1999 book. And so there are a couple of figures that I think that triggered him during that, talking about that. So the first one is this four-dimensional geometry of the development of a regulating organism. So we've talked about regulating organisms that are where the cells send signals to one another, and it determines the fate of the cell. So in C. elegans, we have what we call mosaic development, which is where the cells have a deterministic fate from the mother cell to the daughter cell. That information is passed on, and then by and large, those cells will retain that fate ultimately in adulthood. So the fate is determined from the birth of the cell. In regulative development, however, different cells, depending on their gene expression state, depending on their neighbors, if you take stem cells, for example, and put them into a heart or a liver, they take on the heart, some heart or liver fate. And so this is a regulative embryo. So this is where we have this map. This axis here is developmental time. So these slices going up is increasing in developmental time. So this is the one cell egg. This is the thing that gets fertilized. And then it moves up to this blastula, which is where the cells in the egg divide. They're still pretty much undifferentiated. Then you have, in this next slice, you have what they call an expansion wave and a contraction wave. You start getting tissue differentiation, and then you get these waves that occur in the cell that sort of define the limits of that tissue and work to drive differentiation. And then in this final picture, there's a continuing gene cascade which drives further differentiation. And so you can see that you can build a tree out of this. If you flip it over, you can build a tree where each tissue starts to, you get like an undifferentiated mass of cells, and then you start getting branching events every time a new tissue forms. So some cells start forming a new tissue. There's some gene expression changes in each cell. The communication between the cells or the spatial restriction of cells in a certain area of the embryo define this new tissue, and it branches off from a precursor set of cells. And then you end up with this embryo with many different tissue types. So that's one. And the hypergraphs actually that I've been working with actually look something like this. There are a lot of similarities here. And then this is another example where you have developmental time. You have this, again, the egg. This is mosaic development. So this is like C. elegans development, where the mother cell determines the fate of the daughter cell just by virtue of passing on its genome. So this is the 3D, this is the egg, this is the single cell, and then you get two different cells. They have different sizes here. This is an asymmetric division. So we did a paper on this where we looked at, in the C. elegans lineage tree, we looked at the different sizes of cells, and it's an imperfect thing to approximate because cell sizes are not, you can approximate the size by segmenting images and calculating the volume of that image, but it's not a perfect way of doing it. In any case, you can differentiate between symmetric divisions where the two daughter cells are basically the same size, and then asymmetric divisions where the daughter cells are of different sizes. And so that's what you see here in this slice, where the daughter cells are two different sizes. And then in this slide at the top, you have this you have a continuation of this process of asymmetric division. So when you get asymmetric divisions, you set up these differentiation waves, and you set up the potential for tissue differentiation. Now, because it's deterministic, there's no mystery as to what the tissues will become, but the alignment of the cells and the migration of the cells are actually the thing that's going to matter in this case. At least I think so. But in any case, this is the difference between the two types of development. And I thought these are interesting visualizations because they do show this process over developmental time, and these differentiation waves, and then how they relate to some of these network embeddings that we're creating. So I just wanted to make a point about that. I also have these various images. Okay, this is a book, The Nature of Complex Networks. This is a book that I found interesting. This is actually something, I think it's from this book that I just showed. And this is the evolution of research topics on complex networks. So I like this visualization because I'm really a fan of a lot of this bibliometric stuff. But it does show some of these topics. Some of them we don't talk about in the group much, but some we do. So this is specific to network theory, and it just shows like the frequency of these different topics over time from 1998 to basically the present. So there have been hypergraphs and cyclical complexes. We talked about that. I talked about that with Jiahang on Thursday, actually. So that's been increasing. Artificial neural networks has been increasing even more so. And then this one here, epidemics and spreading phenomena. Unsurprisingly, this has really peaked in the last couple of years for obvious reasons. You have some of these things like small world networks and preferential attachment, which was really big maybe 20 years ago with respect to like small world networks and social networks and things like that. The work of Barabasi and some other people, Strogatz and Watts. And so you can see that there was a trend. You can see a trend towards that maybe 20 years ago, and it's decreased since then. And so there are all these different networks embedded in metric spaces. This is another thing that we kind of talked about in the group. This is kind of grown maybe about 15 years ago and stayed constant. So there are all these topics, and it's kind of interesting how they've been sort of evolving. And then finally, this is a picture of a set of comparative embryos, just to show you the size of different embryos. So this is the axolotl embryo. This is the embryo that Susan and Karan and Harikrishna are working on. It's a very large embryo, as you can see. And if you compare that with Xenopus and lamprey, you see that it's much bigger than Xenopus and lamprey. Xenopus being smaller, but still sort of in the same family as axolotl, but then lamprey is this what they call phylogenetic outgroup, which is it's in the area of like fishes in that area of the tree of life. So this is a much smaller embryo. I don't know if there's a correlation. I don't think there's a correlation between embryo size and phylogeny. It's just that sometimes you get really big eggs in some species and smaller eggs in other species, and but this is just a size comparison here of these different types of embryos. So then I'm going to get into some papers here. Actually, I want to talk about this paper. This is a really nice one. And I'm going to zoom in on it a little bit if I can here. It's down here. Okay. So this is a paper, and going back to this idea of networks, and we've talked about networks in embryos, and we're talking about making these graph embeddings, and we're talking about different hypergraphs and things like that. This paper is about endothelial cell signaling. So this is within the layer of cells called the endothelium, And this is a cell signaling study of these cells. And they say, the title says, Small World Connectivity Dictates Collective Endothelial Cell Signaling. So in the endothelium, you have these cells, and they have signaling between them. As I said, in regulative development, you often see the cell signaling serve to organize things. So it organizes collective behavior. So they're proposing this idea of small world connectivity being the driver. So this is the significant statement here. The endothelium is the single layer of cells lining all blood vessels and acts as a central control hub to regulate multiple cardiovascular functions in response to hundreds of physiological stimuli. So this is an important part of the body, important function, physiological function. The detection of various physiological stimuli is distributed in spatially separated sites across the endothelium. So this is where you have the detection of stimuli by cells. Cells have this sort of perceptual aspect to them that we don't talk about as much as we probably should, but they can sense things in physiological states of the organism. They can sense environmental states. So they respond in kind. And so the distribution of this is spatially separated in different places. So you don't have it, not every cell is equally exposed to these signals. Distributed sensing is difficult to reconcile with the requirement for coordinated cell activity across large regions of the endothelium. So this is basically, you need to have coordinated cell activity, but not all locations in this collection of cells has equal access to the sensing. So what to do? Here we show that the endothelium resolves the issue by using a network with scale-free and small world properties. So scale-free and small world properties are where you have these things called hubs, which are highly connected nodes. And those highly connected nodes serve to distribute information or to aggregate information. And so this is an important concept. Scale-free and small world just refer to their relative connectivity.
Hi, Bradley.
Yeah.
I believe some of that is physical, especially if cells, endothelial cells, are like the epithelial cells on our tensegrity object. So their connections to physical connections to each other also relay information to the cell. And so I just wanted to say that this is partly physical.
Right. Yeah.
And I do have to go now. So
okay. Well, thank you for attending. So this is these cells are connected in different ways, but they're also connected at different strengths. So some of the cells serve to distribute information. Some of them just kind of receive them on the periphery of these networks. And this is these types of organization. This is what they're proposing. The organization confers a high spatial propagation speed and a high degree of synchronizability across the endothelium. The network organization also explains the robust nature of endothelial communication and its resistance to damage or failure. So some of these small world networks, these scale-free networks, which are different types they have just different types of organization. Small world networks tend to have a shorter, what they call path length, and have maybe a more hierarchical organization than scale-free networks, but they basically allow for this type of, this type of architecture. So one of the things that people have pointed out is that in all sorts of networks like this, from communication networks to technical networks to social networks, that they have this resistance to damage and failure and this robustness overall. So this robustness of function. So this is interesting. And so they say network organization controls system behavior by determining the overall intracellular signal propagation speed, the robustness of the system to failures and attack, and the degree of synchronizability in the system. So they use graph theory to sort of get at these issues, and they have a nice, I can't seem to load the rest of the file, but this is basically what they have to say here. I think it's an interesting study in that they're able to, they extract these networks from physiological data, and they're able to characterize these networks and say something about the how the sort of the function. All right, so then if we go down the rest of this paper, we'll see that they have a figure here. And so this figure actually shows some of the things that I'm talking about with respect to networks. So this is a random network, and they usually use this to calibrate some of the things that they're, when they compare scale-free and small world networks, they have to compare it to something because the structure deviates from random in some way. And so the scale-free, this is an example of a scale-free network. And they usually characterize in terms of the degree distribution, or how many connections there are into each node. And so they did this as an average, or they do this over a distribution. And so this is the example here, which is usually scale-free networks as well as small world networks exhibit long tail behavior, which means that there's a long tail of the distribution. So some nodes have more connections than others. In this case, you have a random network. The random network does not have a long tail. It has this sort of random distribution where you have a mean number of connections and then a small amount of variation, less or more than that. So the random connection really doesn't have much structure as a result. Messages get passed from cell to cell, more or less. You have very few long-range connections across the network. In a scale-free network, however, you can make it across the network quite quickly by just passing messages from a node to a hub and then to another node. And so that's the advantage of these scale-free and small world networks. But if we look at this up here, we have it on a lattice. We have, this is in terms of this path length measure I told you about. So in a regular network, you have this Manhattan, what they call Manhattan distance, which is where you move, from node to node in a linear fashion, in a stepwise fashion. So you end up with these path lengths that require you to always go from node to node. There are no shortcuts. In a small world network, you have this structure where you have shortcuts between nodes. So the path length is much shorter. So I go node to node in this case, then I have a shortcut to this node over here, and then a node to node connection here. And so you can travel farther across the graph with a shorter path length. With random networks, you get this shorter path length, but that's largely an artifact of just randomness. So the path length, there is no real advantage to path length in a random network. You get, sometimes it can be shorter, sometimes it can be longer, but there's no structure in the random graph that allows you to have a shorter path length. Then they have a measure called the clustering coefficient. So this just allows us to see like what neighbors are sort of in clusters together and what neighbors are not. And so in small world networks, you get clusters, but you also have these hubs that allow for the easy facilitation of messages, of signals. And so this is the advantage of this type of network structure, again, where you get shortcuts that form and you get one pair among neighbors. So it's, in a random network, you get really non-specific connectivity. In a small world network, you get very specific connectivity. And this is useful for communication of signals. And then you have these structural networks, these functional networks, and then the correlation between these different networks over time. And so what they're doing is they're extracting information out of their cell population. They're extracting information by looking at the centroid of the cell. They're building edges between the centroids. They're building a functional network, which is the function between the epithelia cells, the relationships between them, and then they're correlating that structure on a functional network. This is a lot like what they do in neuroimaging for the brain. So this just gives us a number of parameters to work with. This is just an example of how you can build these networks from a population of cells and some sort of the properties between, say, like a regular connection regime, a random connection regime, and then this middle ground, which are these small world or scale-free networks. So this is an example of some of the cell, like what they're doing with cells. They're measuring, they're looking at signaling itself. So these are the things that define the connections, these signals. And so in this case, they're looking at calcium signaling. They're looking at the intensity of the signal between cells, how they're exchanging calcium and how they're communicating with one another. So that's a nice picture of that. In this case, they do network analysis of acetylcholine and histamine-evoked calcium responses in the endothelium. So they're looking at neurotransmitters, and they're looking at these calcium ions, and they're looking at their activity in the epithelium. And this is another way you can characterize connectivity. So if you have cells that are, activated together, this is like in neuroimaging. If the cells are activated together, if the signal shows up in cells in different places, they can be assumed to be connected. And so this is how they're building this connectivity map. They're looking at this cell culture, where they're looking at the cell population. They're looking at the intensity of the signal for this specific marker that they want. And then they're assuming that that consists of a correlation matrix. So when cells are expressing a certain amount of this marker together, then that's a connection, and it's a strong connection depending on the correlation. And so you can correlate these over time as well, because these processes are time dependent. For example, you don't want to just pick up a spurious correlation and assume it's a connection between two cells. You want to have a stable connection over time. And so this is some of the things that they're doing here. Then here's another example. Here's a network analysis summary. So they're looking at this local structure. They're looking at the community structure of these networks. So the difference between the two, local structure and community structure in this case, is that you have local structures where you have these little modules or these little local networks or subnetworks that form. This is why they're interested in clustering, because sometimes you have clustering among cells, and this can be very advantageous if you want to understand why there are differences across population of cells, that subpopulations are acting differently or that they're clustering in different ways. And so this is important in networks as well, where you have these little network clusters. And you have connections between the clusters, so they're not independent, but they're also not homogeneous across the tissue or across the cell population. So you have these opportunities for differentiation of little bits of function. You have this opportunity to connect different parts of the network and so forth. This local structure shows that you can have these cells that are highly connected. So in this case, you might have a bunch of cells that really highly express acetylcholine or have the calcium ion activity, and then other areas that don't. You can also have areas that are sort of linked together in time. And so there are all these different possibilities. Now, community structure is different, because community structure is where you're finding local communities, and they're broader than these like local clusters because they're connected components. So you have these local hubs, for example, on a small world network, that are connected to its local cluster. And then you have these connector hubs, which connect the two local hubs. And this together forms a community. So you can think about this in terms of hierarchical signaling. If you have a connector hub, and then you have your local hubs, those local hubs are going to be organized by the connector hub. And these clusters or subnetworks are going to be organized by the local hub. And so that may be very important for organizing some of these signals, the diffusion of information, and so forth. So then this is again some of the clustering stuff that they've done with respect to the sensitivity of acetylcholine and some of these other components, and the differences between the random network and the control. And this is just more staining work that they've done showing some of these different things that they've looked: histamine active cells, acetylcholine active cells, and the overlap between them, so you can merge the images and then understand better what these networks are doing from that. So this, I think, is a nice paper. They kind of pull a lot of information out of these images. It's certainly not something that most people do for some of these systems, but I think it's a useful tool to sort of understand what's going on, especially over time. And so that's that paper. So so I guess Longhui had to leave as well, and Susan, thank you for attending. So if we have any other comments, that'd be all for this week. Did we have anything else we want to talk about, or? All right. Talk to you later.